All right, Gino, can you hear me?
I can, Rachel.
Okay, do I sound all right?
You sound wonderful. I think you could go. I think we're
Excellent.
on.
We are on and ready.
Yeah. Go ahead, Rachel. I'm on standby.
All right, perfect. So welcome everyone. A recording and slides from today's webinar will be posted um, at the link that you see on your screen. So hello again, and thank you for joining us today for the third webinar in our EPIC series on the use of NAMs in risk assessment. This series is being co-organized by the Environmental Protection Agency, PETA Science Consortium International, the Institute for In Vitro Sciences, and the California Department of Pesticide Regulation. I'm Rachel Bruner, a biologist with the US EPA, and I'll be co-moderating today's webinar, along with several others, including Gino Scarano, a senior advisor from the New Chemicals Division of the EPA Office of Pollution Prevention and Toxics, Niladri Baumik, a toxicologist in the Human Health Assessment Branch of the California DPR, Amy Clippinger, president of the PETA Science Consortium International, and Christy Sullivan, the vice president of education and outreach at IIVS. Before I introduce our speakers, please note that the slides and a recording from today's webinar will be posted online at this link shortly after the webinar, as I said earlier. All future EPIC webinar materials will also be available at this link as the series continues. Okay, so we have two speakers today who will be presenting on the development and application of NAMs, workflows and tools. First, we'll hear from Elijah Petersen from the National Institute for Standards and Technology, and second from Corie Ellison from Procter & Gamble Company. We will do a Q&A session at the end of the second presentation, and the webinar is scheduled for 1.5 hours, and we may wrap up earlier, kind of depending on how long the questions go. Everyone is muted, and you can type any questions in the questions module within your Zoom toolbar at the bottom of your screen. And please, if you could just indicate if your question is directed to a specific speaker. And we just ask that you don't add questions to the chat. We have also enabled the closed captions, and you can turn this feature on or off according to your preference using the toolbar at the bottom of the screen. All right, so now Gino Scarano will share some introductory comments from EPA.
Thank you, Rachel. Hello, everyone. I see there's 168 of you online right now. So welcome to EPIC webinar number 3. Rachel and I both work in the Tosca New Chemicals group here at EPA, and we have the privilege of co-hosting and working with our colleagues at the PETA International Science Consortium, California DPR, and IIVS to plan this webinar series. I again want to especially thank PETA and IIVS for doing all of the logistics for this important webinar series, which we are committed to bringing to you 4 times a year. And so this is number 3, the first of 2024. So like you, I'm excited to hear from both Elijah and Corie, and I turn it back to you, Rachel, and thank you.
Thank you, Gino. Our colleague from the California DPR, Niladri Baumik, will also have some comments to share now.
Hello and good morning, everyone. We at California Department of Pesticide Regulation are excited to be part of this webinar series on NAMs, and in which this is the first one for 2024. And I extend my warm welcome to our speakers today, and looking forward to the 2 great presentations on ICCVAM validation and building confidence in NAMs. And thank you, PETA and others involved with, for organizing this series again. And back to you, Rachel.
Thank you, Niladri. Okay, so speaking first today will be Dr. Elijah Petersen. Elijah Petersen completed his PhD at the University of Michigan in environmental engineering in 2007. Then he completed postdocs at the University of Joensuu in Finland. I probably said that um, poorly, on Fulbright scholarship, and then the University of Michigan before joining NIST as a National Research Council postdoctoral fellow. He became a staff scientist at NIST in 2010 and works in the Cell Systems Science Group in the Biosystems and Biomaterials Division. His research currently focuses on the development of robust, reproducible in vitro test methods, and he is a co-chair of the ICCVAM validation group. Welcome, Elijah, and please go ahead. You can see your screen.
Okay. Well, thanks again to the organizers for this opportunity on behalf of myself and the whole ICCVAM work group, or validation work group. We're really happy to share about what we've been doing, and hopefully this will stimulate interest to look at the whole report and to go into more of the details. So again, this is a report, or this presentation will be sharing the efforts from a whole group. This is certainly by nowhere even close to my efforts alone. A whole group of us came together, as I'll be sharing in one of the upcoming slides, to put this together. So first off, I'll say a little bit about what ICCVAM is. Many of you probably already aware of this. It was put forward by an act of Congress back in 2000. NICCVAM is a branch of the NIHS that supervises ICCVAM. And so this, what I think is what really cool about ICCVAM is that we bring together the research and regulatory agencies at the same table. Often, as someone on the research side, I can be wondering, how would a regulator potentially, how would this impact them? Because it's something they could use. What are some of their considerations? And so by having the research and regulatory agencies at the same table, it's extremely informative and hopefully, and I believe very much synergistic, as we're able to feed off of each other and learn from each other to help develop these methods to support the 3 Rs. So as I was saying, this was very much a group effort. You can see that there was a lot of people involved, including some people who had, were involved for some time and then took a different position, or for some reason had to leave the group. I'll highlight my 2 other co-chairs, John Gordon from CPSC and Susie Fitzpatrick from FDA CIFSAN, Nicole Kleinstroyer from NICCVAM was also, did a lot of coordination here. In addition to our support staff, Amber Daniel was excellent. Dave Allen did a lot while he was at Innovative. So, there's, I guess, overall, there's so many people who've contributed to this that I really want to acknowledge, the value of all these different perspectives coming together to try to put forward this validation report. So, this is the title of the report. It's about validation, qualification, and regulatory acceptance of new approach methods through our ICCVAM work group. ICCVAM does have currently an old report from 1997 talking about validation. For those of you who've been involved in this space, you can imagine what's changed since then. 25 years later, amazing progress has happened in this area, and we wanted to update, and to reflect some of the current thinking, across different, the federal families for, what are some of the underlying principles. As I see this report as a bit like an umbrella, as what we all could agree upon together, from different federal agencies. And a lot of agencies have also put forward their own, specific guidance, and even centers or groups within an agency. This report, similar to the previous one, is much in line with OECD, again, it's document 34. And we also introduced some new things, such as a context of use terminology, and you'll see that popping up all throughout my presentation. Well, part of that is emphasizing that the validation process is flexible and adaptable. Based on your context of use, you may have different things that you need. And there's a real need for communication between method developers and regulatory agencies and among the federal agencies, and having these work groups kind of brings everyone together to talk through these things as a group. This report is very much following in alignment with the strategic roadmap that ICCVAM put forward in 2018. Some of the key focuses of this roadmap was it helps end users, guiding their development. What we don't want to happen is someone is spending all this time, money, and effort to develop a new approach, and then they haven't had as much contact as they could have with some of the end users, and then end users are left not entirely sure how to implement it or if it was going to fulfill their needs. So part of this is that, as I was saying again, this efficient, flexible approach to developing these new methods, and we want to encourage the adoption of these methods by having the discussion all along the way among different federal agencies and with different stakeholders, such as the method developers. Hopefully, this helps everything work more smoothly. And again, there's no one size fits all. There are many different contexts of use among different agencies and even centers within an agency. So it's really important to be laser focused on what's the specific use and what's needed for that use. This is just one example where you can see within just 2 agencies, there are a bunch of different centers with different types of products that they're regulating. And so what works for one center may not work for another center because, again, the different contexts of use and different needs. To put, another way of putting some of these ideas is that we're transitioning a bit from an old model where you have a validated NAM, have the stamp on it, and then there's kind of a hope or expectation that different regulatory agencies are going to be able to use it. Instead, it's now more about evolving confidence and identifying the context of use, where instead of having centralized like the validation groups where like ICCVAM or ECVAM or what have you, putting their stamp on it, now there's a bunch of different end users and they can, things can develop in different fashion. There's also a movement away from having one method, one new approach method that's designed to directly replace an animal test or provide the piece of information that test was perceived to provide. And now often there could be multiple approaches that are being used synergistically in, for example, a defined approach or something like that. So this report builds upon a lot of recent efforts, or I guess the OECD guidance document 34 isn't so recent, but it certainly builds upon that, which was published after the initial ICCVAM report in 97. There is, as I'll highlight, a revision to OECD guidance document 34 that the U.S. is co-leading, and a lot of the people involved in the developing of this validation report are involved there. There's also been some important work defining the standardization of defined approaches that really can support regulatory use. And there was recently a paper published in 2022 by van der Zalm et al. Talking about a framework for scientific confidence, and many co-authors are shared between the development of this paper and our ICCVAM validation report. I'll highlight too that there was another report that has been published, I'd say more in parallel with the ICCVAM validation report. The ICCVAM validation report came out in August of last year, and there's a National Academy of Science, Engineering, and Medicine report that wrestled with confidence and some similar and some complementary ways, I would say, as someone involved in both of these efforts. And that came out last June or July, I believe. So that was, certainly, that was, there's both of these efforts that are hopefully, leading to establishing these frameworks for scientific confidence. One of the key figures from the van der Zalm et al. Paper shown on the left, where they talk about some of the different key aspects that are needed to build, that are needed in this framework for establishing scientific confidence. And I'll go in the upcoming slides through each of these in more detail. There's also, you'll see on the right side, a slide that or a figure from Ellison Harold's presentation about confidence at the EPA NAM conference that was now about a year and a half ago. And you'll notice that there's a lot of overlap and similarity between these two. So this is the figure for that I'd say is really a foundation for this report. And if you want to read the draft report that we had out for public commenting that went out last August, there's a QR code and a link down here on the bottom right. So, again, at the center of this, we want flexible, fit-for-purpose names for validation. How do we get there? Well, there's these different components: biological relevance, data integrity, technical characterization, information transparency. And then kind of underlying a lot of those is this context of use and independent review, where the NAM is validated or is evaluated across some set of criteria. And again, this figure is adapted from the Van der Zalm et al. Paper that came out about back in 2022. So let's go into each of these topics in a little bit more detail. So context of use, it's, in a lot of ways, there's similarity between that and the fitness for purpose. Again, we're borrowing a figure from the Van der Zalm et al. Paper. So one of the questions that are key is what regulatory statute specifically is a name intended to help comply with? How will it be used as a standalone, as part of a defined approach, as part of a weight of evidence? What is the context in which it's being used? There's very different criteria for if something is used as a pre-regulatory screening prioritization or for quantitative risk assessment, where for quantitative risk assessment, the bar may be a lot higher in terms of what's demanded of it. For example, and then is the information provided sufficient to address the regulatory endpoints of interest? This relates to both the biological relevance and the technical characterization of the method. So this biological relevance question, sometimes it's called human relevance, but within ICCVAM, we do have ecological testing. So sometimes the species of interest is a fish. So as I'm describing this, you know that you're really referring to the species of interest. So we're comparing the biology of the species of interest and that of the test system, and that could relate to things such as the dosimetry modeling, the structure of the organ or tissue, the cells used, and if there is human data, to what degree do the results from a particular method or combination of methods relate to the human response. And so, one of the ways that this can be done is to try to link up the biological relevance of another method that's believed to be biologically relevant with that of the new method being developed. Historically, this has often been done through comparing results from a NAM to an animal test. And we've specified in the report that we shouldn't just assume that the animal test is going to relate the best to human biology or the mechanism of toxicity. And it's important to look at it objectively on a case-by-case basis to understand to what degree does the animal model capture or not capture some of the biology that's relevant. And when we are using benchmark animal data, it's important to look at to what degree, if you have the data, do they relate to human effects? And to understand the variability of the animal data and to characterize that so that when you're comparing, and taking that into account when you're comparing data from a new approach or set of approaches to the animal data. So it's important to consider the accuracy by looking at the consistency across methods and approaches, the ability to get, what responses you get for positive and negative reference chemicals, and to really have this focus on the biological relevance of the species of interest. So we do highlight in a table in the report some endpoints where there's been demonstrated biological and mechanistic relevance of NAMs for supporting regulatory applications. And and you can see that there's references, often to EPA documents or OECD documents, and some of the endpoints where this has already been demonstrated is skin sensitization, endocrine disruption, developmental neurotoxicity, and inhalation toxicity. So now I'll get into the area where I do most of my work, which is on the technical characterization. Here, the focus is on understanding the source of uncertainty and variability in the assay. You may want to evaluate what's the applicability domain, or of course, you will want to evaluate that to understand to what things does this assay work and to what context of use does it not work. For example, some assays where there's adherent cells at the bottom of a 96 well plate, it may not work to add a nonpolar solvent to it because there's solubility issues, as just one example among many. So you want to characterize the reproducibility within a laboratory. A transferability may be needed depending upon the context of use. Sometimes you need it, sometimes you don't. How the assay, the reproducibility of the results for reference chemicals and control measurements, and its limits of detection or quantification. As you report, reporting data from a method, you need to share what protocol and equipment you're using and computational models, if any. And what is, again, this whole context of use question, what is considered acceptable may depend upon its intended use. So for screening assay, you maybe have different thresholds of what you find acceptable as compared to something that's used for a quantitative, for example, quantitative risk assessment. One of the figures was borrowed from a paper that I published in collaboration with with people from different ICCVAM agencies and also some international collaborators. I'll briefly walk through, this figure. So let's assume we have a scientifically relevant NAM as a kind of a first step. And once we have that, we describe, in this paper and throughout, and then also in this report, some of the conceptual evaluation tools that could be used, such as ways to plot out the expected source of variability, the assay design, and what control measurements you're incorporating, and check sheets to support fulfilling GLP or GLP-like criteria. Then, once you have that done, you can move on to testing within the laboratory, and that could include looking at the performance across time with control charting of your control measurements, looking at robustness testing to assess what is the parameter space in which this assay works acceptably, and looking at interactions among among the control measurements to ensure that they're independent of each other. This can then all feed into statistical analysis so that you could potentially have, in addition to whether the assay is a positive or not, a statistical confidence associated with that. For some assays, you may need interlaboratory testing. That's not always needed. It really depends, again, on your context of use. And then if everything goes perfectly, you'd have a high quality, robust method. But in practice, I've never had this experience. I don't know if anyone else ever has, where you just kind of march through these steps and you're done. Usually it's an iterative process. So as you'll notice, these dotted lines on the right-hand side indicating that you may be doing the interlab testing and you have to go back and do more robustness testing. For example, if one of your laboratories was, for some unexpected reason, getting results that are a bit different than the range that was observed in the initial laboratory, you may want to assess, well, does that make a difference? How much flexibility is in this method? Maybe we, need to see if there's more flexibility, or maybe one of the protocol steps needs to be revised to clarify something. So often it's really this iterative process, and at the end you can lead to a robust method. Data integrity and information transparency is key. We want to have a clear linkage between the raw data that's generated and that in the final report to avoid mistakes that could happen along the way. We want to have communication about the method so that people who are interested in learning more about it could go into some of the details. Through the testing I just described, you can talk about the limitations and uncertainties, and it is often valuable and necessary to have independent reproducibility of the data, such as looking at the prediction models, the processing of the raw data, and so forth, especially if there's a statistical model used that may be necessary. And if there's proprietary parts, there's some considerations that the OC has put forward that may be a way to work with that dimension. This is a list of the reports in the tables in the report. We specify some, as I was saying earlier, this is kind of like an umbrella, and then within that, different agencies may have put forward their own guidance so that you could look to get more information about the specific context of use for that agency or even a center within the agency. One of the things that ICCVAM does, I think, is really a great contribution is to go in and understand really for a specific endpoint, how are agencies approaching this? How are they using the data from different methods to try to fulfill a certain testing need? So by having the regulators at the table in addition to the research agencies, often we can have the right discussions and provide some insight into how that could be done. And so that's really important as you're developing these for method developers to understand how might my method be used. And as I've already described, we have a table for the biological mechanistic relevance of NAMs and how and documents that have and for endpoints where that's been already discussed. So in summary, really we're trying to get to this increasing scientific confidence. We want to have the model clearly identified with a clear context of use. I want to show concordance with the species of interest or across different methods. Want to have the information publicly available and to the extent possible reviewed by independent third parties. And we want to have the biological relevance for related to the human biology and the mechanism of toxicity well defined in addition to the technical aspects. So this is where we're at now and where we've been in terms of a timeline. We started this back in March of 2021. It was collaboratively developed by all participating federal agencies. Again, it was definitely a team effort, and every agency involved brought their unique perspectives and needs to the table and experience. We had a draft that was put forward for commenting back in August 10th. We got a lot of excellent feedback. And we also got excellent feedback from the CDPR meeting in September, where they weighed in and provided some additional things to think about. The document is now undergoing final review by the participating agencies, and we anticipate the report to be released in early 2024. So I'd say we're nearing the final stages. One of the really interesting piece of feedback we got from the public commenting and the CDPR was trying to have a forum to link the regulators and method developers. And so we're hoping that this can really help support, making that linkage again between, identifying the context of use and having this kind of dialogue. So we're gonna hope to have every 2 to 3 of these a year, having this forum where we're trying to proactively highlight the recommendations of our report, and provide, again, this interaction between the NAMs developers and the federal agencies and other end users. So we're going to focus each time on a specific endpoint. The first one will be on carcinogenicity testing. And we're going to have as a pre-recorded webinars where industry and regulatory stakeholders can chime in about their needs for that endpoint. And then during the forum, the method developers can then share, here's methods we're developing, and here's why we believe this is fulfilling some of the needs within this context of use. And with that, that's happy to pass it on back to the next to either Rachel or Corie. I don't remember who goes next. Rachel, you're next, and I'm happy to answer any questions. And thanks again for this opportunity.
Thank you, Elijah. That was a great presentation. We are going to hold all questions until the end of the next presentation. I'm sure we have questions, but if you do have them, you can add them to the Q&A module as they come up. And again, note if they're for a specific speaker. Speaking next will be Dr. Corie Ellison. Corie Ellison received a PhD in pharmacology and toxicology from the School of Medicine and Biomedical Sciences at the State University of New York at Buffalo. Dr. Ellison is a principal scientist at the Procter & Gamble Company in the Central Product Safety Division, where his current research efforts are in the areas of pharmacokinetics, absorption, distribution, metabolism, and excretion, and new approach methods, particularly in addressing how findings in these fields can improve human health risk assessment for chemicals. So thank you for being here, Corie, and please go ahead. And I can see that your screen is being shared.
Great, thanks, Rachel. And thanks, Elijah, for the good talk. So as stated, my name is Corie Ellison, and I'm a human safety toxicologist at Procter & Gamble. And the title of my talk today is Establishing Confidence in New Approach Methodologies, Use of NAMs to Refine and Strengthen SAR Read Across for Branch Alkyl Carboxylic Acids. So a mouthful. I first want to thank several groups, including my P&G colleagues who contributed towards the work that I will present today. Cefic, who funded the research that I will present, and the EPIC webinar organizers for inviting me to speak today. So starting off here is an outline of the case study that I will present. So I'll start by explaining the aims of the case study, then present the NAM data that was generated to address our questions related to toxicodynamics and toxicokinetics. I will then end the presentation with a summary and discussion on establishing confidence in NAMs, the NAM approaches used in the case study. So the case study that I'll present today focuses on branched carboxylic acids. And one of the most well-studied branched carboxylic acids is valproic acid. Valproic acid is a widely used anticonvulsant. However, valproic acid is also a known teratogen, which can cause developmental effects in human and animal models. And 2-ethylhexanoic acid is an isomer of valproic acid and has been shown to also be developmentally toxic in rodent models. And there's been a number of different studies where various branched chain carboxylic acids have been tested in a variety of screening level developmental toxicity assays. And there's an apparent trend that some of these branched carboxylic acids cause valproic-like developmental toxicity effects, while others do not. So thus, there is sufficient information to indicate that some analogs of valproic acid can have developmental toxicity potential. However, it's not clear whether the active compounds act via the same mode of action, or if there are limits of structural similarity beyond which biological activity diverges. So as the focus of the case study was to investigate whether a group of branched carboxylic acids, as pictured here on the slide, had the potential to have valproate-like effects on development. For the purpose of the case study and presentation today, I'm going to use the EPA definition of a NAM, or a new approach method, which is shown here at the top of the slide. And our selection of NAMs for inclusion in the case study was determined by a hypothesis-driven approach where we gave consideration to the problem statement that we were addressing and the applicability of a given NAM. We ended up using 4 main groups of NAMs, which were toxicologically based structure-activity relationship read-across, transcriptomics, in silico docking or in silico molecular docking, and in vitro and in vitro and in silico toxicokinetic approaches. So an underlining element of the case study is this concept of read across and trying to better understand where it is or isn't appropriate to use for the current case study chemicals. So read across is an old concept that's being increasingly utilized for chemical risk assessment and regulation. The basic premise for read across is that a compound with similar chemical structure will have similar biological activity. That's either because they interact with the same target or because they are metabolized to a common intermediate that acts as the active compound. And the use of read across requires identifying quality analogs and demonstrating that the analogs have similar activity. So for the case study, we are interested in understanding if the case study chemicals, which are the branched chain carboxylic acids, have valproate-like effects on development. So this slide shows a very high-level overview of the NAMs that were used for the case study and how each serves a specific purpose for informing the SAR read across. So I'll go through each in more details on the coming slides, but first wanted to just show this general illustration of the case study work so you can see how the NAMs complement each other, but at the same time, each NAM brings its own independent data stream. So the toxicogenomic data that we'll present or that I'll present provides information on gene expression patterns and also possible mode of action. The molecular docking data provides information on possible interactions between an enzyme binding pocket and a chemical. And the pharmacokinetic data provides information on internal exposure and dose equivalency between the chemicals. And when taken together, they can all help inform the SAR read across and help us understand which of the case study chemicals should be grouped with valproic acid because they are expected to have valproate-like effects on development, and which chemicals should not be grouped with valproic acid because they are not expected to have valproate-like effects on development. So the transcriptomic studies were done for all the case study chemicals, and we utilized a study design that had 4 different cell types and 3 doses and an exposure of 6 hours. And for each in vitro exposure scenario, which consisted of a given chemical, dose, and cell type, the top up and down regulated genes were identified and used to create a transcriptional signature. That's what you see here. And the transcriptional signatures were then compared among the different case study chemicals, so the different branched chain carboxylic acids, to determine similarity and differences between the case study chemicals. And additionally, an additional analysis was done, which is known as connectivity map analysis or CMAP analysis. And CMAP analysis involves comparing a chemical's transcriptional signature to a large database of existing transcriptional signatures. So you can see if your chemical of interest, if the gene expression pattern is similar to anything that's already been tested within the database, which has 300 plus chemicals. So the figure on this slide represents what the CMAP analysis for valproic acid looks like. And to get such a figure, we first had to determine the transcriptional signature for valproic acid, and then query the transcriptional signature of valproic acid within the CMAP database, and then see what chemicals in the database have very similar gene expression as valproic acid. And chemicals that have a gene expression signature that closely match valproic acid get ranked at the top of the list as shown here. And they show up in blue for this particular figure. And the close connections between valproic acid and a given chemical in the database is shown by this bright red kind of square box. So where you see a lot of red, a lot of bright red, means that valproic acid is acting or has a similar gene expression signature as these chemicals in the database. And the CMAP analysis showed that valproic acid has a very similar gene expression pattern as histone deacetylase inhibitors, also known as HDAC inhibitors. So we also did this CMAP analysis across all of our case study chemicals and observed several different results. So first, the transcriptional profile of valproic acid, which is VPA, ethylhexanoic acid, and propyl nonanoic acid were all highly correlated. You can see where all the red kind of aligns. And also these chemicals had transcriptional signatures that were very similar to HDAC inhibitors. And then lastly, we saw that some of the other case study chemicals shown here across the graph had transcriptional signatures that were different than the HDAC inhibitors and also different than valproic acid, ethylhexanoic acid, and PNA. And as a result of this data, we began to investigate whether HDAC inhibition could be associated with valproate-like effects on development. And if you go into the scientific literature, there's literature to support the idea that HDAC inhibition can induce specific malformations, and there are multiple toxicity studies which have evaluated different HDAC inhibitors. A common mode of action among these chemicals is histone hyperacetylation and the consequent alteration of gene expression. Additionally, HDAC knockout mice display a phenotype that resembles teratogenic activity. So given this information, we were next interested in using molecular docking studies to determine if any of the case study chemicals would be expected to be ligands for HDAC. And if the molecular docking results were consistent with the transcriptomic results. So this is the structure of HDAC. It has been previously determined by others. That's available in the literature. And as such, it's possible to conduct molecular docking studies to determine if a chemical would be a good ligand for HDAC. So on the left-hand side of the slide is the structure of HDAC3. And then on the right-hand side is a close-up of the binding pocket of HDAC. And the binding pocket, excuse me, the binding pocket has two asymmetric binding pockets. So one small binding pocket and then one large binding pocket. And in this particular figure, we're also showing how valproic acid would sit in this binding pocket. And it turns out that valproic acid is expected to be a good ligand for HDAC. So we also reviewed the other case study chemicals that we had in this particular case study to see if they would be ligands for HDAC. And results from the docking studies show that valproic acid, which is shown in green here, 2-ethylhexanoic acid, which is shown in pink, it's the pink chemical, and propananoic acid, which is PNA and shown in yellow, were all expected to be good ligands for the HDAC receptor. So they fit well within the binding pocket of HDAC. However, the rest of the case study chemicals were not considered good ligands for HDAC, but for different reasons. So PHA and HDA were expected to be poor fits for HDAC binding because the chemicals were too bulky to fit into the binding pocket. They actually extend outward. And then MNA, OA, and EBA were expected to be poor ligands, but because they were too small to fit well within the binding pocket, they would just kind of fall out. So what we ended up seeing, and I'll go to the next slide here. So using the results from the transcriptomic and docking studies, we began to develop structure activity relationships for the branched alkyl carboxylic acids. And we're able to group them into different groups based on mode of action. So depending on chemical structure, some of these chemicals can fit into the HDAC binding pocket, but there are certain structural requirements which must be met. And a chemical is expected to be teratogenic if the R group has 2 to 4 carbons, and the R1 group has 3 to 7 carbons. So based on our assessment, the branched alkyl chain carboxylic acids that do not meet these criteria would not be accepted to have valproic-like effects on development. And you can see those here, and many of them being the longer chain acids. So in addition to evaluating the toxicodynamic effects of the case study chemicals, we were also interested in the toxicokinetic effects. And we wanted to better understand if there would be differences in ADME or toxicokinetics, and whether similar administered doses would result in similar internal exposures. And to answer these questions, we studied a subset of the branched carboxylic acid, so 2 of them, so VPA and EHA, which are expected to be developmental toxins, and then PHA and HDA, which we expect to not have valproate-like effects on development. So the toxicokinetic-based NAMs that we use are shown on this slide. We use in vitro and in silico approaches to help answer these questions. The in vitro assays that we use were CACO2 permeability to understand the potential for oral absorption. We used in vitro equilibrium dialysis assays to measure the amount of plasma protein binding for a chemical. And we used in vitro hepatocytes to measure the rate of metabolism. And once we have the different pieces of in vitro data, we use physiologically based pharmacokinetic modeling as a way to integrate the data and get estimates of internal exposure. So as a summary, the chemical-specific ADME data is shown on the slide. So the permeability rate through KECO2 cells was very similar across the chemicals. High permeability was observed, and this indicates that the chemicals are expected to have relatively high oral absorption. The plasma protein binding data show that valproic acid and ethylhexanoic acid are higher fractions unbound compared to the other two case studies that we examined, so PHA and HDA. And then when we look at the rate of hepatocyte metabolism, the in vitro hepatic metabolism data show that HDA and pentoheptanoic acid are metabolized at much faster rates than valproic acid and ethylhexanoic acid. So similar to the toxicodynamic data, the in vitro TK data is showing that the longer branch chain carboxylic acids are behaving differently than these shorter chain branch carboxylic acids. So now that we have the specific ADME or the chemical specific ADME data, we are interested in understanding what the impact of the different ADME parameters would have on in vivo kinetic profiles of the chemicals. So to investigate this, we developed an oral rat PBPK model, and this would allow us to integrate our in vitro ADME data and make estimates of internal exposure for a given chemical. And the modeling approach would also allow us to establish oral equivalent doses for each chemical. And shown on the left-hand side is just the general workflow that was used for the PBPK models, and I'll go into further details on the next couple slides. So for each chemical, we used the physiology of a rat, and we were modeling an oral exposure route. And the chemical-specific parameters related to oral permeability, plasma protein binding, and hepatic metabolism came from in vitro data that I showed a couple slides ago. And then we also had some in silico estimates for log P, water solubility, pKa, and partition coefficients. And then when it was possible, we ran simulations from the model and compared them to existing in vivo PK data sets, so we could evaluate the performance of the model. And a representative example of the model performance for valproic acid shown on this slide, where the line represents the model simulation, and then the symbols represent the experimental in vivo data. And the model estimates for valproic acid and for ethylhexanoic acid were in good agreement with the in vivo PK data. So this gave us confidence that the current modeling approach is suitable for the branch chain carboxylic acids. We then use the PBPK models to estimate the internal exposures that would result from a common oral dose to rats. The PBPK model for each chemical was set up to simulate an oral dose of 100 milligrams per kilogram, and then the plasma concentration from the simulation was recorded. And this is shown in the figure here on the slide. And for a common dose, the longer chain carboxylic acids result in a lower internal exposure, and this is due primarily to the faster rate of hepatic metabolism that we saw with the in vitro data. And what this means is that when trying to achieve equivalent internal exposures, the longer chain carboxylic acids must be given a higher dose. So building off of that, we then used the PBPK models to run a final set of simulations. And this time we're interested in establishing oral equivalent doses across the different branch chain carboxylic acids. And to estimate or to establish an oral equivalent dose, it's first necessary to pick a target internal exposure. In this case, we were interested in using the internal exposure that was associated with the developmental NOEL for valproic acid. So we first set up the valproic acid model to run a simulation at a dose of 150 mg per kg, which is the developmental NOEL. And when running the simulation of 150 milligrams per kilogram, the resulting Cmax for plasma in the rat was 128 micrograms per milliliter. So this plasma concentration of 128 micrograms per milliliter then becomes the target internal exposure for the simulations that we want to do with the other branch chain carboxylic acids. So if we move to the middle of the slide, this shows what external dose was needed in the heading here, was needed in order to result in an internal exposure of 128 micrograms per milliliter in plasma. And what we observed was that the longer branch chain carboxylic acids have a higher oral equivalent dose compared to the shorter chain branch carboxylic acids. So to summarize this, I know I have shown a lot of data and I'll try to make sense of it here. So to summarize, for the branch carboxylic acids, the toxicokinetic NAMs indicate that as the branch chain length increases, the rate of hepatic metabolism increases. This then caused the longer chain chemicals to have lower internal exposures to the parent chemical. The PBPK modeling indicated that the longer chain branch carboxylic acids would require about 3 to 7 times higher oral doses to result in a similar internal exposure as valproic acid. On the toxicodynamic side, the NAMs showed that substituents on the carboxylic acid have a significant impact on the developmental effects. As the branch chain length increased, the developmental effects of the chemicals appear to decrease. Valproic acid, ethylhexanoic acid, and PNA had similar gene expression patterns as well as HDAC inhibition potential and could be considered suitable analogs for developmental potential. The other case study chemicals have very different gene expression patterns when compared back to VPA, EHA, and PNA, and they also did not show HDAC inhibition potential. So these chemicals can be considered as very weak or not having developmental toxicity potential. And overall, we use the toxicokinetic and toxicodynamic NAMs to show that not all branch carboxylic acids are expected to behave similarly. And as such, different analog groupings should be considered when doing a toxicologically based SAR read-across assessment. And you can see how we suggest the binning based on these two boxes below. So this slide captures some of the ways that we were able to establish confidence in the current NAM approach. So the NAM was hypothesis driven. The selection of NAMs was based on our problem statement and our understanding of the biology and was fit for purpose. Additionally, the data from the different NAMs all converged to a common answer and were in agreement with each other. The chemistry and the biology aligned across multiple chemicals and allowed us to derive structure-activity relationships and mechanistic understanding. And with respect to read across, there's guidance from regulators and scientific articles that explain how to do this in an objective and scientific manner. Additionally, there are frameworks which aim to estimate the confidence associated with a given read-across assessment. And then this being my last slide for today, I thought it'd be interesting to try to map this NAM approach to the figure that Elijah just shared earlier from the ICCVAM report. So keeping in mind that the ICCVAM report is really focused on a single NAM assay, but some of the concepts apply to what I believe an overall strategy, NAM strategies I present today. So the context of use that Elijah talked about could be established via the case study problem statement and is a hypothesis-driven approach. The biological relevance of the NAM approach was established by using biologically based NAMs, a hypothesis-driven approach, a mechanistic understanding of the data. We used reference data and compared to legacy animal data. Data integrity was ensured via multiple independent checks of the raw and processed data throughout the case study. Technical characterization was achieved in the case study by using state-of-the-art approaches with appropriate controls, reference chemicals, analytical methods, statistical models, and documentation. And then as far as information transparency, all of our data was published. And then as far as the independent review, this approach has been submitted to TOXI and was accepted for publication via the traditional review process. So with that, I conclude and just want to thank you for your attention and time, and I'll turn it back over to the organizers.
Great, thanks very much, Corie, and thank you, Elijah, for your presentations today. We do have time now for questions for both of our speakers. And so thanks everyone who's typed them into the Q&A panel, and please do continue to pop them in there. So I'll just jump right in. First question is for Elijah, and when I read it, I'm seeing kind of a two-part question here. It's about validating, ICCVAM validating or perhaps vetting NAMs and how scientists from other countries can be included. So maybe it's worth talking a little bit about sort of what ICCVAM does or doesn't do with regard to validating or vetting NAMs, and then how others can be involved.
So, I mean, ICCVAM is, for the U.S. federal agencies, I'd say there's a number of ways that there are nevertheless connections internationally. Certainly international harmonization is valuable for many reasons when possible. There is like a ICCVAM group. It's I don't remember what the exact acronym is now, but there's basically different validation groups internationally have discussions and try to get on the same page for certain topics. So there's that's one way. There's international connections. When a method is going through a standards organization or OECD and the like, there's certainly international discussions at that point. Some of the and there may be on for the validation management teams, we may have international representation. So there's I would say a goal and an intention to have international connections. When possible, recognizing that it's also a U.S. group. This is as just as ICCVAM member. I'm I'm not the co-chair or Nicole or someone who has a much broader vantage point than I do, but that's how I view that. But for the validation report, certainly international harmonization is something that's aspired to, and that ICCVAM is engages in different ways, I would say.
Yeah, great. Great, thanks, Elijah. And I would maybe just add on to that there is the ICCVAM Public Forum every year in May, and it's coming up soon. It's May 20th and 21st. And if I'm not mistaken, it's usually a hybrid meeting, so it would be pretty accessible to folks around the world.
A good point, as is the Science Advisory Committee meeting that happens in September. And often we have some international people involved in that. So I'd say we're having those discussions.
Great. All right, Elijah, I will stick with you for the next one as well. The question is, will the MDFs be open to regulators outside of the US? If so, how would those regulators express interest in being invited?
I don't know. I can pass along this inquiry to Nicole and some of my colleagues in ICCVAM to ask about this. I don't know if we've really considered international regulators. I think at some point, obviously, that would be happening. One of the, from my perspective, one of the tricky things is different regulatory agencies have different flexibilities in terms of interacting informally with test method developers. So I'm not entirely sure to what degree other international ones could if they had wanted to. But since we do aspire to international harmonization, it's certainly something to consider, and I'll be inquiring about that with my colleagues.
Oh, great. And I probably should have mentioned that. I think that comes from one of our colleagues at Health Canada, so can pass along that information.
I will indeed. It's a fascinating question, and I don't recall that we've discussed it yet, but I will be passing that along. Thank you for inquiring about that.
All right. Next question is an interesting one, and I would say this could be for either of the speakers, or maybe even for some of the panelists and hosts here. Gino, Christine, Naladri, Rachel, feel free to, whoever would like to tackle this one. So with respect to scientific reliability and regulatory agencies' adoption of new methods, does this group ever consider legal reliability of new methods? So meaning courts and judges need to accept the viability of new methods in order to find scientific evidence legally reliable. So has anybody sort of thought about that legal topic?
Yeah, I'd be happy to chime in and then open up for others too. I appreciate this because it reminded me of something I meant to mention but didn't in my slides, which is the validation report does not bind anyone to anything. It is just it was providing suggestions and advice, but no one is bound to it. I think with topics like this, it really gets then to the next level, which is agency specific input. So different regulatory agencies probably have different. My guess is they may have different takes on this question, and so I think with the agency specific guidance, they may address as they're also considering some legal issues and other things. I assume I'm not in one of those agencies, but I would assume they would. There probably is more relevant guidance there. But I'll step back, and if anyone else would like to chime into it, welcome that.
Well, I'm certainly not from an agency either, but I can mention that the ASCCT actually had a webinar about this about 2-3 years ago, where we had some legal experts who frequently testified in court cases related to toxicology, and it was a very, very interesting conversation. It was a very popular webinar, and I can paste the recording in the chat for people to take a look at. But I think, to me, this raises the question, we're always thinking about who are the audiences or communities that we need to reach out to talk about new emerging methods and how they can be used. And this says to me, experts, how are judges making these decisions? They're advised by experts. And so are these experts kept up to date on new models and methods and the data coming from them? Gino,
you're
Sorry,
muted.
I was muted.
There we
I
go.
know. Thanks for the questions, and hi, Elijah, and hi, Corie. I was going to mention, Christy, that exact webinar, which was amazing. I forgot the guy, but he was a toxicologist and a law professor at Arizona, Gary someone. You're muted, but anyway. Was it Gary?
Gary Marchant, yeah.
Thank you very much. It was very good. I wanted to mention two things. Number one, under the Toxic Substances Control Act, the amendments that were made in 2016, which added Section 4H that talked about reducing testing in vertebrate animals. It has a legal definition that I think we've been struggling with ever since, which gets at the legal liability issue, and that is in developing NAMs, they need to be scientifically reliable, relevant, capable of providing information of equivalent or better scientific reliability and quality. And so, I think that begs that question. And I think that is important. The second thing, and I might put you, Christy and Amy, on the spot because I'm no longer as involved, but have NAMs been discussed in the OECD arena under the mutual acceptance of data consideration? Because I think that is an international way to get different governmental authorities across the world to accept information that's generated in one country or another. And I'll stop there.
Yeah, I mean, there are certainly test guidelines built from in vitro assays or combinations of in vitro assays. So those are clearly under mutual acceptance of data. There's also a lot of discussion, of course, now, well, about how to incorporate in silico modeling within that new concept as well. And the new defined approach test guideline has some in silico modeling now under MAD. So those conversations are definitely ongoing at the OECD level. But yeah, thanks. This is a really great question, and I'll add the link to the webinar into the chat.
Great, thanks everybody. All right, so there's a number of specific questions, Corie, to your talk. So I'll run through these here. Why did you choose Cmax instead of AUC for the comparison?
Yep. So Cmax was chosen because for valproic acid, they've actually looked probably in the mid 80s to see if it's Cmax driven effects or AUC. And they did this with a bunch of animal studies and maybe even some of the human data. And they were able to determine that was Cmax. So you could have similar AUC exposures and not see some of these effects. But then once you get to Cmax and tying that to the effects, it was really driven by Cmax. So it's more of a peak concentration rather than total area under the curve. So that's why we went with Cmax.
Thank you. Let's see, for the in vitro metabolism assays of the 4 compounds, female rat hepatocytes were used. Do you see any gender differences in vitro, and are there any species differences in vitro?
We didn't specifically look at gender differences. There's probably something published, at least for valproic acid. The reason why we went with female was because we were going against a developmental endpoint and knowing that it was in utero exposures that were most likely going to be the ones that then impact the developing fetus. So we said, well, that's going to happen in the female. So we went with female hepatocytes.
All right, and then how do these strategies address long-term exposures and cumulative impacts of multiple exposures, and how does that affect how risk assessment is done?
Long-term exposure. So there's like 3 different questions in there. So let me see if I can bring it up. So long-term exposure. So generally with risk assessment, long-term exposures can be handled in a number of different ways. You can try to have your animal studies be multi-generational animal studies or be of chronic duration, and that will help you understand long-term exposures. If you don't have that, then oftentimes in risk assessment, you would then use an uncertainty factor, which can cover for duration. So again, valproic acid's one where it's been looked at in a number of studies going from acute to long-term. And then on the developmental side, it's really more to do with the critical window of developmental, of fetus development. So you really want to make sure that your exposures are happening during the critical window for development of the fetus. So that's where it's probably less about long chronic exposures for the endpoint that we were looking at. And then you said mixed exposures, or was there another part of that question?
Yeah, cumulative impacts of multiple exposures.
I guess I would need to clarify if that's coming from one chemical or multiple chemicals. So right now we are focusing just on a single chemical where the cumulative exposure in vivo could again happen multiple times, and that's how we typically look at it. with animal data showing daily exposures. And then within the in vitro system, we chose to look at, for like the toxical dynamic, or for the gene expression data, we chose the 6-hour time point, not because we weren't interested in repeat exposures, but because we were interested in direct acting effects of the chemical. So in that case, we chose a shorter timeline to make sure that we're catching the effects that were a direct result of our test chemicals.
All right. Here's a good question, maybe a little bigger picture. So I'm curious about how much emphasis is placed on addressing the identification of assumptions and how they are accounted for or documented. So in Corey's example, chemical structure was assumed to be sufficient for defining similarity. And then an assumption is also that the reference database has sufficiently similar and relevant chemicals. So what scrutiny was placed on proper assembly of the database or on defining these background details?
Yeah, so P&G, we are strong advocates that chemical structure alone is insufficient to identify suitability. So chemical structure is a starting point, but then we also give emphasis to phys-chem properties. We give emphasis to metabolism, pathways. This is where we brought in the additional biological data to not just look at chemical structure, but to look at biological activity. So that's where, and then also the different ADME properties. So that's where a lot of times people will just look at chemical structure. And that's kind of what the first slide or two was, where if you just look at, hey, these are all branched chain carboxylic acids, let's default to valproic acid. They almost act like that because they're branched chain carboxylic acids. And the real main point is we're not doing ourselves any justice with that. So really, that might be a starting point, but then to look at different data streams, whether again, it's metabolism pathways, whether it's toxicogenomics data, to then better understand how that compares and where we see similarities, differences, activity cliffs, et cetera. And then the database, so that's a BRO database, so that's not housed or maintained by P&G, but it's something that's open to anyone that does work with toxicogenomics. So it's a strong database to move away from structure. So the the HDAC inhibitors that we found were not based on structure at all, but it was based on more mode of action and what biological targets we were hitting. So that's because I was looking at the gene expression signatures. So for those that are getting into toxicogenomics and identifying gene expression signatures, you now have a way to look at biology similarity rather than just structural similarity. So that's where we think read across should be cemented in, and then not just trying to use a simple structural similarity score, but there's a lot more that goes into it.
All right, there's still a number of questions, so I'll keep reading them off here, Corie. So if there are conflicting values for the no adverse effect dose levels, is the assumption to take the worst case scenario?
So again, a big question. I'm sure people on the screen have opinions on this. So the value I picked was not identified by me or P&G. It's one that was identified by those in the 80s and 90s that really studied valproic acid and many different studies. I will say it's not uncommon to get conflicting no L's, low L's, points of departure, and toxicity studies. And it's kind of what Elijah said. There's no one answer or one way, like there's no one NAM. There's no one way to address this. You want to look at the data. You want to look at study quality. You want to look at species sensitivities. You want to look at many different things to then pick what is the appropriate critical study for your test chemical. So it's not necessarily defaulting to the lowest numbers, not defaulting to the highest, but following the science, following the data integrity All those things that Elijah had on his screen for what makes for a good NAM, that also exists for animal studies. And we need to make sure that we're actually looking at the data and not just a summary number that people publish.
All right. How were the cell lines selected for the toxicogenomics, and how was the cross-species element considered? TD in humans, TK in rats.
Yeah, sure. So cell lines were selected. So we were going after, I think it was 4 major organs. There was breast, lung, heart, and liver. So those were the 4 cell lines. We've done, we and others have done work to try to understand what's the right number of cell lines. There's differences of opinion for how many cell lines you need. In this particular case, what we were trying to do was hit on a number of different cell lines which have been shown to be active. And in particular, we have previously shown them to be active to valproic acid. So we know that our test chemical will be active in these cell lines. And then one of the other things that we were doing was not trying to cover all biological space and understand everything that's being hit, but to try to understand similarities and differences within our case study chemicals. And there's, again, been previous work that's been published that show that you can use a relatively small battery of cells to answer that question. And you can see whether you get similar gene expression signatures in those cell types at common doses across your battery of chemicals. So that's how we assess that. And then for the cross-species extrapolation, so TD, TK, it's a bit of a mix, I understand. So part of it was, and the mode of action, we know that valproic acid is a human teratogen. We wanted to understand some of the human mode of action, which is why we use the cell lines there. What we don't have are some of the Noel, we don't have human Noels. We have that in the rat. And we know that if we are anchoring to legacy animal data or legacy toxicity data, it's going to be animal data. So that's where on the TK side, we were looking to say, okay, if we gave similar oral doses in a rat toxicity study, would we really expect there to be equivalent internal exposures? Would we expect the Noel to be higher or lower for one of these branch chain acids if we did a new tox study? So that's where it's kind of a mix. And then a final risk assessment, which this isn't. This is more about a read across and grouping. A final risk assessment should give consideration to a given data stream, understand what species it's coming from. Are there need for uncertainty factors to cover interspecies differences or within population sensitivities? So that would come at the final stage of when you really pick what your critical effects are, your critical data, and then determine if you need to account for any uncertainties across species.
Great. 3 more currently. So
Yeah.
thanks for hanging in there. These are really great, great answers. Yeah, thank you. All right, so let's see. Okay, on one of your slides, you stated teratogenic if C1 is less than R is less than C5 and C2 is less than R1 is less than C8. Assuming you remember which slide that is. Does this number account for the total carbon number, or is it the backbone chain length?
Yeah, I'm going to show this.
Okay.
So we have a great medicinal chemist. We actually had 2 great medicinal chemists help us understand this relationship. So the R is this part of the molecule. So this R here is corresponding to here, and then the R1 is corresponding to here. And with the R needing to be 2, 3, or 4 to be teratogenic, and this other one being C3, 4, 5, 6, or 7 to be teratogenic. And it really goes back up to here, where if you remember, there's 2 asymmetric binding pockets for HDAC. So the one smaller R group needs to be big enough to anchor itself in, because the really, really small branch chain carboxylic acids, like this guy here, are too small to really fit in and interact with the binding pocket, and they kind of move out easily. So it's not so much the total number of carbons. I think the question was, but it was given for this R group of the branch carboxylic acid. It needs to be within the 3, 4, or 5, 3 or 4, 2, 3, or 4, and then on the larger side where there's a larger binding pocket is where you can extend between 2 to 8. So hopefully that clarifies the question.
Yep, thank you. That's helpful. Thanks for bringing up the slide.
Mm-hmm.
All right. And then next one, did you look at any chiral interactions at the binding site as it relates to exerting toxicity? So as an example, the R enantiomer of EHA is active for teratogenicity, whereas the S enantiomer is inactive. Curious of your thoughts on chirality's influence on toxicodynamics.
The broad we, looked at this. Me, I did not, but we did look at the chirality, and our, again, medicinal chemist that looked at this, his name is Gong. If you want to look at the author list that I had on slide 2, he would love to talk to you about this if you want to follow up offline. It's beyond my my comfort zone for talking here, but I can tell you we looked at it and he's got some thoughts about how things sit within the binding pocket and why you'll see the differences with the correct chirality. And I'm guessing it goes back to that slide I just shared and how well they fit into that binding pocket.
All right, great. And then last one that's up so far is a question, did the gene expression data show other toxicities outside of the development risk?
So I believe when they did the CMAP and gene expression analysis, they were really docking, and I don't want to misspeak here. So again, you can send this question to Jorge Nassif. He's the one, another author that really looked at this. I know that none of the longer chain acids hit anything that was an HDAC receptor. I believe they didn't hit anything else that was developmental tox associated with it, but I don't want to speak out of place here. So again, you can follow up with an email if you want to get a clear answer, because I'll probably just say something wrong in the moment.
I doubt that, but sounds good. All right, so that is all of the questions that were submitted. Thank you again, both of you, so much, and thanks, Corie, for pointing people to the paper with the co-authors. I'm sure if folks reach out to any of us here on the screen or the co-authors listed on that paper, we'll make sure we get questions to the right place. So yeah, anyone who has a question that they didn't want to ask today, feel free to follow up with any of us and we'll get it to the right place. All right, and so again, thank you so much to both of our speakers. Thank you all for joining us today, and please do keep an eye out for our future webinars in this series. It's a quarterly webinar series, so our next webinar will be in May, and it's going to focus on some developments in the eye irritation testing space. All right, thank you all so much. Have a great day, and we'll see you next time.
Thank
Bye.
you, everyone. Great questions. Thank you.
Bye.